Welcome back. What we've done so far is that we have split the reads into reads belonging to the mids for 3 different samples. And then for each of those samples, we have split the reads into reads belonging to each primer for each exon of each HLA gene that we're interested in, HLA A, B, and C. So in total now you would have HLA A, B, and C times 2 exons, that's 6, times by forward and reverse primers, that's 12 different files. So now you can see just by looking at the file that this is a lot more uniform than what we started off with, as you would expect. And when I click on the dots, you can see that it's actually very uniform, but a few things stand out to me, such as in position 60 here, we have a column of predominantly Gs. Similarly here, another G column, another G column, another G column. Over here, a couple of A columns and a T column. If I examine position 60 in detail, I can see out of the 50 sequences we had the master sequences has reported a T at this position. An A has been recorded here, and then we have 48 reads which are recorded a G. What I would think has happened here is that we have 2 sequencing substitution errors, the T and the A, and in actual fact it should have been a G in this position. And similarly in this position here we have 48 Gs reported and only 1 T and 1 A, so again it should have been a G. So this sequence that we've chosen as the master sequence actually has quite a few substitution errors in it as we look across the page, where it just basically disagrees with almost every other sequence. The second thing that stands out to me is that we have this short sequence here. I'm not sure what's caused that, but in a file where you have plenty of reads to do what you want, probably the short sequence would just be ignored. The next thing that stands out is I have one sequence here which is in agreement with the other sequences for the start of the sequence, but then is basically in complete disagreement for most of the read. And now what has caused this? Generally this would be caused by one of 2 things called indels, insertions and deletions. This sequence here, I would guess, has probably either got 1 or more bases inserted into it with respect to the other sequences, or it has 1 or more bases which have been deleted from it with respect to the other sequences. And I can check that by moving to the edit mode and insert. And then if something had been deleted from this read with respect to the others, I can just replace that by putting a dash in here to show that something had been deleted. But as I put some dashes in, I notice that it still does not line up very well. So I'll just remove those dashes. So if something had been inserted into the sequence with respect to the other sequences, this C perhaps might be an insertion. So I can just delete this C, and then suddenly actually it lines up quite well. And so again, much the same as with the short sequence, if I have lots of sequences for what I need to do, this sequence would quite possibly, we could ignore it in our analysis. But as we've fixed it up, we'll leave it as it is. Okay, the next thing that I notice is that if you remember, this primer had a mixture in it at position 3, I think it was. And that primer was had an S or a C and a G mixture. But also I see mixtures occurring further across the page. I see a TG mixture here versus CC. And where there's the T and the G there, if I go across, I see that I also have a G in this position, and I have a GA in this position. And similarly, where there's TGs here, I see they have that G and that GA. And so there seems to be 2 patterns of reads here. And what these 2 patterns represent are sequences which are taken from the exon 2 of HLA-B from the 2 different chromosomes. So this is a human sample. We have 2 chromosomes. And basically HLA-A is the most variable region within the human genome, and we see that this person has two different sequences for exon 2 for HLA-B. And so what we want to do, we call those two different sequences two alleles. What we want to do is that we want to find the consensus sequence for each of those alleles, for the CC allele, we'll call it, and the TG allele. And the best way of finding those consensus sequences is to group the reads for each of those alleles together. And we do that exactly the same way as we did previously. We go to the sequence menu and we sort by the sum residue frequencies in the selected columns. So we see here are the CC alleles and here are the TG alleles. Now, one thing which is difficult to see at the moment, because this sequence we have as our reference sequence has quite a few errors in it, is may become visible if we choose a different reference sequence with less errors. So I'll just choose the second one down. And now we notice we don't see any columns down the page. This is a much better read, doesn't have nearly as many sequencing errors. And you can see now that you can see where those columns of G's, where we have a T here and a T here and a T here. So now what might become evident with a bit closer look, if we look, this is obviously the first difference between the two alleles, the CC versus the TG. And if we scroll right over to the other side of the page, we'll see that the last differences between the two alleles are this GACCA. As the GACCA lines up with where the TG was, almost, but not quite. If I go up the top of here, I can see I've got a GAC here, another GAC. This one's got a CA, I can see that here. And if I come back down to where the TGs were, I can see this one doesn't have the GACCA. It also actually doesn't have the G here. So what's happening here? Well, this is not a sequencing error. This is something which has probably occurred in the PCR. And these reads represent reads which have part of one allele and part of the other allele. They're what's called a recombinant. And it was probably formed during the PCR process. And I won't go through any details for that because you probably would have gone through that elsewhere. But it is important to be aware when you're looking at the sequencing data that you can have reads which are a combination, if they come from PCR, that are a combination of different starting material, the two different alleles. And it's important to recognize that you wouldn't obviously use these GACs when you're working out the consensus for the first allele because they don't really belong there. So how do I work out the consensus? Well, the first step in working out the consensus is to actually work out which part of the read is to be included in the consensus. And you shouldn't include the mid in the consensus because obviously the mid is not part of the human genome. And also you should not include the primer in the consensus because the primer sequence does not have to be an exact match for the part of the human genome which it overlay when it joined on. And in fact, we can probably see that by just looking at this GC position here. Was there a G or was there a C at that position in this actual subject that we have sequenced? I don't know, but we know we had primers with Gs and Cs, and whether we amplified with the primer with the G, we got the G, and where we amplified the primer that had the C, we got the C. But what the subject had, we can't really tell from here. So we have to make sure that we start our consensus sequence after the primer ends. So if we go back to the primer sequences, I can see that the primer sequence for HLA-B exon 2 forward ends with CCT, CT. And if I look here, there's the CCT, CT. And so therefore I can start my consensus position and my consensus at this position here, which is the G. And because the read that we've chosen here fortuitously has no sequencing errors, well, the consensus sequence should be the same as this read sequence. until we get to this position here, 4 bases from the end. And when we get to this position, 4 bases from the end, we see this one has a C, and there's a few other reads that also have a C, and there's quite a few reads that have an A, and then there's a few reads which have a G. So basically this is a position that it's difficult to be 100% sure what the consensus should be at that position. And again, fortuitously for us, it's only 4 bases from the end, so I think that we'll just end our consensus sequence at that position. So we go back to the start of the read, we click just before the G, scroll to the end, hold down the Shift key, and again we click just before those last 4 bases, just before it, and copy that. We then go to our text editor, start a new file, and we're going to make FASTA sequences for each of these consensuses. So a FASTA sequence starts with a greater than sign and then the name of the read. So this is mid2, HLA-B, exon 2, forward, and I'm just going to denote this one with an A for the first of our 2 consensuses and paste that in. And then we would do the same for mid2, HLA-B, exon 2, forward, and we'll just denote the second one as being B. So we'll go back and we just choose one of these ones, and randomly I've managed to choose one that doesn't seem to have any sequencing errors. But what I'm going to do is I'm going to try and find one that does have a few sequencing errors, just when you're doing the other samples, you may well find that you can't find a read that has no sequencing errors. So to show you how to deal with that, we don't want to choose a read that has that many sequencing errors. Let's see. It's hard to find one. I want to find one which has just got a couple of sequence errors. Oh, there's got a couple of columns of A's. But yeah, so there's 2 obvious sequencing errors here. There's an A, these 2 columns of A's. So we basically, this one is largely correct apart from those 2 columns of A's. So what I'll do is, remember we start our consensus sequences in this position here. I'll just click there and hold down the Shift key, and again we just want to end. This one's a little bit shorter, so it's 3 bases from the end. Copy that. Go across to the PS pad and paste that in. Go back. Now we need to fix up those errors. So first thing we need to do is find where the errors are. So if I just copy that piece of sequence, and you see it starts about 68 bases in, but remember that we lost approximately the first 30 bases for the mid plus the primer. So basically this should be just under 40 bases in when we find this in the file. So if I just find that, okay. So you see that there is where our 2 errors are. So what we'll try and do is that we'll just try and, this sequence here you see, which is a few sequences down, it has no errors in that position. So what we'll do is that we'll select from there to there. So this is, oh come on. This is what the correct consensus sequence should have been in those positions. I'll just copy that and paste it on top of where the error was. So now this here is an exact match of what our consensus should be. It's basically we've used the part of 2 reads to combine to make the consensus. So what I would now do is I would repeat this for exon 2 reverse and create 2 more consensuses, and exon 3 forward and reverse, and then I would repeat it for HLA A and C and for the other mids. And then once you have all of those, you can move on to the next step. Thank you.